Okay, so hello, I'm Holden, and my partners are Audrey and Erin, and today we're going to be discussing lipid rafts. So, just to provide a quick overview of where we're going to be going with this presentation, we're going to start with some background, and then we're going to discuss the structure and formation of lipid rafts, along with their specific functions and pathology. And then we're going to go into some clinical applications and relevance, and briefly conclude. So, So, as just some broad background on lipids, lipids are a chemically diverse group of fatty compounds which consist of a hydrocarbon chain and a terminal carboxylic acid. They're categorized based on their individual properties such as solubility, reactivity, function, etc., and they perform a wide variety of functions within the body, including signaling, energy storage, and acting as structural components of cell membranes. So, on the topic of cell membranes, membranes are instrumental for the organization and signaling of cells, but in order to actually be functional, they have to be dynamic and comprise a fluid pattern. So, membranes are made up of a wide variety of components, which differ extensively from one to the other, often consisting of phospholipids, sphingolipids, cholesterol, integral proteins, and carbohydrates, among others. We use something called the fluid mosaic model to illustrate this defining concept. And then on that topic, sterols or cholesterols are a specialized type of lipid containing four fused rings. They're unique and dynamic in their fluidity in that they alter membrane fluidity at different temperatures. At cold temperatures, they increase the fluidity. At warm temperatures, they decrease the fluidity. And then some more specific background on lipid rafts. So, lipid rafts serve as platforms for the spatial organization of membrane proteins. Proteins associated with these rafts often possess specific lipid modifications, which facilitate their selective localization within these microdomains. They do feature elevated levels of cholesterols and sphingolipids. These rafts are dynamic and ordered. They're very tightly packed compared to the surrounding membrane, and they are crucial for cellular signaling processes. They act as hubs for signal transduction by concentrating signaling molecules and receptors, thereby influencing the efficiency and specificity of cellular communication. Over the years, extensive research has been conducted to understand the roles of lipid rafts in membrane organization and cellular signaling, unraveling their impact on various physiological and pathological conditions.
Okay, so getting into the structure of these lipid rafts, I'm going to start by saying they are they're nanoscopic. And so because of this, a lot of the research and knowledge that we have on these lipid rafts were done by in vitro experiments because it is very hard to visualize them in vivo, as well as tagging them with labels reduces their affinity for raft domains, which affects their function. And then there's a high concentration of glycolipids within lipid rafts because they're used to protect the membrane and help with structure. And then there's also a high concentration of cholesterol because they're used to reinforce rigidity and prevent osmosis and leakages. And then we have GPI anchored or GPI linked proteins, which are just peripheral membrane proteins on the outside of the cell that help with signaling and are involved in trafficking processes like endo and exocytosis. And in the schematic, we can see that lipid rafts prefer saturated phospholipids in comparison to unsaturated phospholipids, and that goes hand in hand with the formation of these lipid rafts. We can see that the components within lipid rafts, like the saturated lipids, the sphingolipids, and the spherules, have an attraction towards each other, whereas they are repulsive. They have repulsive interactions with the unsaturated lipids, and as well as there's a preferential interaction between sphingolipids and sterols due to the saturation of sphingolipid hydrophobic tails, but also to the hydrogen bonding between these lipid species. The amide of the sphingolipid backbone can act as both an acid and a base and donate and accept hydrogen bond. And so getting into the function of these lipid rafts, I'm going to start with the exclusion of components. Lipid rafts are lipid rich and protein poor, which entails that they're excluding components. And this has only been shown in isolated systems because, as I've mentioned earlier, it is very hard to visualize these lipid rafts at the scale that they are. But protein depleted membrane domains have been observed in vivo. The direct mechanism is just not known like we know it for in vitro mechanism, like we know it for in vitro experiments. And the exclusion of components goes hand in hand with facilitating cell sorting, because this exclusion of certain proteins is useful in the act of cell sorting because mainly only components that like lipid rafts accumulate in them, being on a basis of aggregating with molecules with similar properties like polarity and solubility, as well as that they may be used to organize subcellular traffic, segregating components and sorting organelles like the Golgi and endosomes to facilitate their traffic to the plasma membrane. And then signaling is also a very important function of lipid rafts because they provide a platform for organizing the signaling of many receptors and proteins, because many of these processes involve inflammation-related pathways, which Audrey will later discuss within the clinical relevance of lipid rafts. And lipid raft composition is carefully regulated. And lastly, protection of lipid rafts. Lipid rafts provide protection to the cell from harsh outer environments like hydrochloric acid in the GI tract. And then the schematic on the right, we can see that some transmembrane proteins rely on Raft affinity for their subcellular localization. Raft affinity is essential as non-Raft mutants are missorted and ultimately degraded. And the endosomal compartment is at the crossroads of vesicular flow between the Golgi and other cellular components. And sorting early endosomes give rise to recycling endosomes, which are in constant exchange with the plasma membrane. And so getting into one of the studies that we observed about lipid rafts, basically it studied 2 different viruses and found that lipid rafts are essential for the transport of influenza virus hemagglutinin, which is also referred to as HA, compared to the virus vesicular stomatitis virus V, VSVG, which does not utilize lipid rafts. And basically the study showed that HA localizes to the apical membrane, whereas VSVG, which does not utilize lipid rafts, localizes to the basolateral membrane. And so within this study, they utilized 2 different kinds of cells. They utilized BHK cells, which are non-polarized cells, and kidney MDCK cells, which are polarized. And that was done to show that even in non-polarized cells, if you're a lipid raft protein, you're going to go into a lipid raft, because in both the non-polarized and the polarized cells, we observed the same mechanism of the HA virus utilizing lipid rafts to be translocated to the apical membrane, and BSVG utilizing a vesicular transport not involving lipid rafts to be translocated to the basolateral membrane. And so in figure 4, we can see that the removal of cholesterol from the BHK cells affects the TGN to surface transport of the influenza virus HA, but not of VSVG, because VSVG does not utilize lipid rafts. And then we see the same thing in figure 5, where the removal of cholesterol from the MDCK polarized cells did affect the translocation of the HA virus, and the HA virus that did make it to the surface was actually missorted and translocated to the basolateral membrane, which is not what it's supposed to do. And we can see that VSVG, which again does not utilize lipid rafts, was not affected by the depletion of cholesterol. And basically the gist is just showing the importance of cholesterol in lipid raft events, and supports that lipid rafts protect against harsh conditions, because the apical membrane is the site of the endothelial cells that interacts with harsh conditions like hydrochloric acid in the GI tract.
All right, and continuing to look at the interaction of pathogens and lipid rafts, there are a lot of studies that have shown that lipid rafts are actually preferential sites for binding and entry of pathogens, such as bacterial toxins and viruses. So as seen in this figure to the right, things like cholera toxins can bind to glycerosphingolipids, and HIV can bind to CD4 receptors, and both of these are more highly concentrated in the lipid rafts compared to the non-raft regions. And then once a virus enters a cell and starts replicating, it might actually take advantage of lipid rafts as a site of assembly and also a platform to bud and exit from the cell. So as seen in the figure, the HIV capsid actually has these things called GAG proteins that can associate with the lipid raft and induce curvature into the membrane and allow it to bud out of the cell and create its own viral envelope as it exits. Additionally, pathogens can even alter lipid rafts in order to decrease immune responses. As Erin mentioned, these lipid rafts are often involved in inflammation signaling pathways and other types of processes. So pathogens can really interfere with this. HIV and lipid rafts this is an interesting topic of study, and in this one primary research, paper, the experimenters were able to look at cell cultures of macrophages, and they used 4 different agents that disrupt cholesterol. And this ends up disrupting lipid rafts. So results showed that HIV, first of all, co-localizes with markers of lipid rafts. And then once you disrupt the cholesterol in those rafts, this can inhibit HIV entry and also lead to less HIV receptors on the cell surface in general. So here's a few figures from that paper. The left is showing the co-localization of the virus with the lipid raft markers. And then these various graphs are 4 different agents that disrupted cholesterol. And the graphs are showing the amount of reverse transcripts created, which is basically the activity of the HIV virus. And so you can see the activity is going down as you add more of the disrupting agents. All right, and then there are a lot of other pathologies and disorders that might be associated with lipid raft involvement. For example, in Alzheimer's disease, there is research showing that lipid rafts might actually be involved in the formation of amyloid beta, which are those toxic peptides formed from the amyloid precursor protein. So a potential mechanism for that is that lipid rafts could help concentrate things like beta and gamma secretases that are necessary for cleaving that protein into the toxic form. And this also might serve again as a kind of platform for aggregation. And this is where they start forming those tangles and end up leading to neurotoxicity. In prion diseases, prion proteins are actually very abundant in lipid rafts because they are GPI anchored proteins. And there is some evidence that the rafts themselves might induce a conformational change that changes a prion from the healthy form to the misfolded form. And then that misfolded one can go on to create this cascade where it causes other ones to misfold. And since they're all concentrated in that area, this could be a potential place where they are interfering with the cell. Then for cancers, disruption in various signaling pathways can lead to unregulated cell growth and tumor progression within the lipid rafts. And defects could also lead to apoptosis resistance. So all of these things are pointing to very important applications and clinical relevance of lipid rafts because they could serve as therapeutic targets. So in the disorders that I just mentioned, oftentimes these are because there might be either too many lipid rafts or increased stability of the rafts, and therefore targeting the rafts and being able to disrupt their stability while still preserving most of the healthy functions could be potential mechanisms for treatment. So drugs that target cholesterol, such as cyclodextrins, can actually deplete the rafts of cholesterol, and things like statins can disrupt biosynthesis of cholesterol. And at low concentrations, these might be able to, again, disrupt the rafts that are pathological while still preserving some healthy function. And then adelphysine is a molecule that might actually help concentrate things like death receptors into the pathological rafts of cancer cells and allow them to undergo apoptosis when otherwise they're resistant. So overall, there's a lot of studies, again, that disrupting cholesterol in rafts could lead to decreased viral infection and decreased amyloid beta, things like that. And overall, lipid rafts could play an important role in targets and for drugs delivered to the cell, just because, again, they're involved in signaling and taking things into the cell.
All right, so just to briefly conclude, lipid rafts are important for many cellular processes, especially signaling. They do utilize a unique organization and are comprised of unique components. They are clinically relevant and could serve as key targets for therapeutic intervention in conditions that Audrey mentioned, such as HIV and Alzheimer's. And then as far as the research is as of now, there is a lack of direct visualization of these rafts because they are so small, dynamic, and transient. But in the future, to overcome this, we could continue advancing microscopy for better imaging of these rafts, and then also perform further research on how these rafts could be utilized as therapeutic targets for the conditions that we mentioned. So yeah, here are some references, and that is it. Thank you for listening.